Dear students, welcome to the course Biochemistry 2. In this course, you will learn the principles of biochemistry and their applications on health and disease. In the beginning, we will discuss the concepts of bioenergetics. As the terminology is self-explanatory, bio and energetics, meaning that we will be dealing with the concepts of energy transfer in biological systems. Generally speaking, the living cells have to perform certain work to stay alive, to grow, and to reproduce. They should have the ability to trap energy and use it to make biological macromolecules, for example, carbohydrates, fats, and proteins. Plants use energy in such a manner that cause them to release oxygen as a waste product. However, human beings and other animals require oxygen as a component to survive. Now, coming to the definition of bioenergetics. Bioenergetics is the quantitative study of energy transductions, which means that it deals with the energy flow through living systems, and the concept of bioenergetics help to understand the nature and function of chemical processes underlying these transductions. All animal and plant cells are powered by chemical energy which is stored in the chemical bonds of organic molecules. Animals convert the chemical energy of fuels into motion and heat, and in few organisms such as fireflies, chemical energy is converted into light. Now, let's take a look at some examples of major bioenergetic processes which are occurring in nature. For example, glycolysis is a biological process in which glucose is broken down into pyruvate and it generates two molecules of adenosine triphosphate, which is the energy currency of biological system. Another process is gluconeogenesis, which is the reverse of glycolysis, in which glucose is synthesized from other biomolecules such as from proteins, amino acids, and fats. Another process is citric acid cycle, which carries out cellular respiration and generates ATP by the oxidation of energy stored in the food material. These processes will be discussed in detail in coming lecture. Dear students, biological energy transformations obey the laws of thermodynamics. There are two fundamental laws of thermodynamics. First law states that for any physical or chemical change, the total amount of energy in the universe remains constant. Energy can be changed from one form to another, or it can be transported from one region to another, but it can neither be created nor destroyed. Before we study the second law of thermodynamics, we have to get familiar with the term entropy. It was first used in 1851 by Rudolf Clausius, and it literally means a change within a system. In general, entropy can be described as disorder of a system. By definition, entropy is the quantitative expression of the randomness or disorder in a system. When the products of a reaction are less complex and more disordered than the reactants, the reaction is said to proceed with a gain in entropy. Entropy is denoted by capital S. When we use the sign of delta with anything, it indicates the change in that particular parameter. Delta S means the change in entropy, and it has a positive sign for a system when entropy increases, and it will be negative when the entropy of system is decreasing. Units of entropy are joules per mole into Kelvin. Now coming to the second law of thermodynamics, it states that the universe always tends towards increasing disorder, and in all natural processes the entropy of universe is increasing. To understand this in a better way, we can take the example of hot cup of coffee, in which heat energy will flow from the cup towards the environment or towards your hand, because heat energy will be more disordered if it will be more dispersed. This is the reason that heat always flows from hot to cold object and not vice versa. The term that describes the heat of a system is known as enthalpy. Enthalpy indicates the heat content of a reacting system. It reflects the number of chemical bonds and their types in the reactants and the products. When a chemical reaction releases heat, it is known to be an exothermic reaction, and value of enthalpy, that is H, is negative. On the other hand, When a chemical reaction is taking heat from the surrounding, it is known as an endothermic reaction and it will have positive values of enthalpy. Enthalpy and entropy of a system are related to each other and they help us to understand Gibbs free energy, which is denoted by G. Gibbs free energy expresses the amount of energy which is capable of doing work during a chemical reaction at constant temperature and pressure. When a reaction will proceed with the release of free energy, the energy change will have negative value and the reaction is known as exergonic. Whereas in case when the system is gaining free energy, then the reaction is known as endergonic and the value of G will be positive. The unit of Gibbs free energy and enthalpy are joules per mole or calorie per mole. Please note that 1 calorie is equal to 4.184 joules. This equation delta G is equal to delta H minus T into delta S indicate a relationship between the free energy, enthalpy, temperature and entropy of a system. In this equation, delta G is the change in Gibbs free energy, delta H indicates the change in enthalpy of the system, T is the absolute temperature and delta S is the change in entropy. And by this we complete our first lecture. We will discuss more terminologies in coming lessons. Thank you.